Hello dear viewers, welcome back again in Glittering Biology with your Bio Guru Alok. Here once again I am along with you with my new lecture related with the chromosomal theory of inheritance under the chapter of principles of inheritance and variation in unit 2nd, that is the genetics and the evolution. Here in hope you just watching the lectures. In the previous lecture, I was discussing about the pleiotropism and the polygenic inheritance, and now it, this theory, chromosomal theory, is related with the same chapter. Here in the chromosomal theory, what we need to study here are Mendel, a very specific name related with the genetics. He was contributed a lot of knowledge for understanding different changes in the characters as well as in the behavior of any organism, and for these specific changes he just denoted a special unit that was considered at that time as a factor, and later on it was just known as the genes. On the basis of Mendel's work, we got the patterns of the inheritance, and by that we are just knowing about the location and functions of the chromosomes nowadays. The role of chromosome was not very specifically explained and during the Mendel's law, but later on, after the 1902, whenever the Sutton and Bovary has proposed the special resemblance with the genes and the chromosomes, then we got a clear cut idea about the chromosomal theory, which is related with the study of genetic material during the cell division. Here we can just discuss about the similarities related with the genes and the chromosomes. What are actually these points are written in your book, that both chromosomes and the genes are found in pairs. That is well known by you all. In the 10th standard you got an idea about this one. And during the gametogenesis, homologous chromosomes get separated, as similarly as the genes also get separate during the gametogenesis during the meiosis. Here the paired condition of both chromosomes and the genes is restored after the fertilization. You got this idea during the meiosis, while we are just studying about the sex determination in the human beings. So on these observations, he proposed a special chromosomal theory of inheritance. What are the important features related with that one? They are just zygote is the diploid cell in the any organism which is formed by the fertilization, and during fertilization zygote receives one maternal set of chromosome from the ovum and one paternal set of chromosome from the parent, means from the sperm. The these two chromosomes of one type, which we are just getting from each parent, together form a homologous pair of chromosome. Then entire throughout the life of the individual, chromosomes retain their individuality and structural uniqueness. Each chromosome carries specific genes which play an important role in determining the characters of the organism. And at last we can say that during meiosis, or we can just relate it with the gamete formation. During gamete formation, the two chromosomes of the homologous pairs get separated from each other. Since factors are located on the chromosomes, they also get separated. The entire concept what we are just discussing right now, that was further explained by another scientist, that are Th. Morgan Alfred H. Struvent and Bridges. The term factor, which was used by the Mendel, was replaced with the term gene by Johansen in 1909. Here, Thomas Hunt Morgan, in the very beginning, I told you he is a renowned person in the field of genetics, and what he has done, he has performed his experiments on the fruit fly, which is very important under the genetics branch, that is Drosophila melanogaster, and provided experimental proof in the favor of chromosomal theory of inheritance. That's why it is very important. And he has selected the entire he has selected this Drosophila melanogaster for this experiment. Why? Because they are growing on the simple synthetic medium in the laboratory, and their life cycle is also very less. So we can just get the several progenies in a short time period. Here, after this, in your book, it is just given about the chromosomal explanation of the law of segregation. You can just go through with this one if you need to get more details related with that, and law of independent assortment. But in the exams, it is only asked that what is the chromosomal theory and what are the special points related with this one. So I'm just taking that much part related with the syllabus. Now the next heading which we need to study, that one is the linkage and recombination. Linkage and recombination. This concept is also not new for you. Actually, you have just gone through with the linkage in the previous 10th standard. What linkage is? It we can just define that it is the tendency of genes which are present on the same chromosome to remain in their original combination and to enter together in the same gamete. For entire observation, here again the Morgan is playing a very important role for proving that, and that process of a linkage. And in the other language, we can say that the genes which are present on the same chromosome remain together as a block. When chromosomes, when chromosomes are getting transferred to the next generation, actually they are transferring themselves in the pair, and this is just known as the linkage. Here, linkage gene, it is the next heading given. You can just look the genes located in the same chromosome being inherited together as known as the linked genes. What is the linkage groups? Linkage groups, what are? All the genes which are located in a single chromosome form one linkage group. The total number of linkage group in an organism corresponding to the haploid number of chromosomes, means to the number of chromosome pair. For example, Drosophila melanogaster has 4, garden pea has 7, maize has 10, and man has 23 linkage groups. I think you have got what is the meaning of haploid, and by that only we can just find out the number of linkage groups. Later on, it is given about the independent assortment versus linkage. What is the uh, difference between these two? We can just say the linkage. In this process, two different genes on the same chromosomes are just found, and if there is no crossing over, that is linkage is 100%, then only two types of gametes are produced. These will be having the specific combination. Like if we have selected capital A and capital B on one chromosome, and on the other homologous chromosome it is small a and small b, then the result will be either capital A B combination or a small a b combination. While in the independent assortment condition, what it will be? We can just say that it is the condition in which the two different genes on two different homologous chromosomes are found, and in this the various combination produced, that may be the four types of gametes. These are, the situation will remain same. Here we can just keep the capital AA combination or the capital B and small b combination, in which you can get that capital AB combination, capital A and small b combination, and small b combination. These genes are present on separate chromosomes. There is neither linkage not crossing over under the independent assortment condition. You can just get a clear cut idea about the thing what we discussed through this figure number 5.17. Later on, it is given about the linkage was first observed by the Bateson and the Punnett. Punnett name is not new. Earlier we discussed about the Punnett square and told you what it is. In 1906, in pea plant, he described the coupling. Later, we will just discuss about that concept of linkage by the Morgan. Here it is given that Drosophila melanogaster, just examined by the Morgan, observed that the genes located on the same chromosomes produce more parental combination than the non-parental combinations. Morgan concluded that coupling and repulsion phenomena of genes are two aspects of the linkage, coupling and recombination. So he defined linkage as the tendency of genes present in the same chromosome to remain in their original combinations and to enter together in the same gamete. This is the another definition proposed by the that Morgan. Salient features of the theory of linkage, what it is, we can just keep these things also in mind, that first point, genes are arranged in the linear fashion in the chromosomes, that you know very well. On the special locus they are present, and genes that show linkage are situated in the same chromosome. Third point, linked genes tend to remain in their original combinations during the course of inheritance. And fourth point, the genes which are closely situated show strong linkage, while those widely separated have more chance to get separated by the crossing over. So these are the special points related with the theory of linkage. What it is about the types of linkage, there are the two types of linkage, complete and incomplete. Just have a look of that one. When linked genes are inherited together for two or more than two generations, the linkage is called as the complete linkage. This phenomena is rare and is found in certain insect species, including male Drosophila. This happens only when the chromosomes does not undergo any breakage, either by crossing over or by some accident. This is called as the complete linkage. While whenever we will just talk about the incomplete linkage, then here we can just say that if two genes fail to remain together for two or more generations, the linkage is called as the incomplete. This generally happens due to the crossing over. And incomplete linkage is very common between the genes which are distantly placed. So here some examples are given. You can just go through for further details related with the complete and incomplete linkage. And I think that is sufficient for today due to the that network issues. We will just discuss the next topic in our next lecture. Thank you everyone.